Hello, my name is Anil Thanki, and I talk about Persist-seq. So it's like it's like infrastructure we developed to analyze the single-set transcriptomics data for consortium that we have lots of collaborators. So this is overview of the consortium. We have so Persist-seq consortium is like a mixture of lots of international institutes or companies, and main aim is to study the all the persistent tumor cell, and it leverages the single-cell sequencing technology, and it generates lots of data sets, and it involves multiple teams or platforms and environments across the different institutes. We need a robust solution, so that's why we came up with a new infrastructure. So one of the to start with, what are the challenges in the modern biology? So modern biology has a high data complexity and the massive data sets, and when it especially in the single cell data that we use. So we need a reproducible infrastructure, it should work across the institutes, not it shouldn't be it works on my system or your system. So ad hoc tools or small pipelines here and there doesn't work out. It's a big bottleneck. And the traditional pipelines are error prone. They need manual intervention, and also the scalability is a big problem for the pipeline. So that's why we need a collaborative setting. When it comes to the setting like ours, we have multiple institutes. This problem just multiplies. So why? This was the reason why we need a new approach. So because we have large scale data sets, and we knew from the beginning because we have multiple institutes sequencing genetic data. Data sets, we have large scale data sets, we need to start a robust solution. It should scale up easily, and it should be automated, scalable, and reproducible, because we need to keep on doing the same analysis again and again. And so it should be also considered across different teams, and it should be minimal manual intervention. So this is our technology stack. So sorry, it's not a standard bioinformatics talk. It's more about the infrastructure. So this is our technology stack. We use Kubernetes, Jenkins, Slack, Galaxy, and AWS. So Kubernetes is it gives us the flexibility and reproducibility to go on any cloud. So right now we use EBI cloud, but we can also deploy this system on the Google Cloud if you want. Jenkins is to orchestrate or automate everything. So Galaxy is a multi-purpose tool here, so it can run our production pipeline, and also it gives a playground for our biologists to play around with their workflows, and also it gives it serves as an exploration tool. They can download the data, or they can play with the data within the Galaxy. And AWS is our scalable storage system that we use across all institutes, and Slack gives us real-time updates, if your workflow fails, or if some sample needs manual intervention. By joining all this stack together, we get a fully automated system that works from data ingestion to the results delivery. It's all automated without any manual intervention. So this is a simple overview of how this system works. So the data pulled from AWS automatically, and Galaxy. The Galaxy libraries are created, and the workflow executes, and the upload results back to the AWS, and also it gives the LS via Slack, so we know the samples are coming and going fine. So this in this whole thing, the Jenkins plays a very crucial role to orchestrate everything. So we have an automated pipeline running 24/7, looking for new samples. It will download the sample from AWS and create a local copy on our Kubernetes cluster. So then it will create the data libraries for Galaxy, and then we run the workflow using something called Galaxy Workflow Executor. This is a command line interface for the Galaxy. So it'll be we don't, because we are using Galaxy, but we don't use the GUI. We use command line to submit all the workflows automatically. So it will fetch the workflow file from GitHub. and the input file from our Galaxy data library, and it will like translate the results, and the results will go back to the Amazon cloud, so our collaborators can download. So like just a little bit about the Galaxy Workflow Executor. Sorry, I forgot the last step. So like this is like automated process, like it goes 24/7 looking for new samples, and like it will repeat the same process for every sample that we get. And like a little bit more about the Galaxy Workflow Executor, like it's a command line interface for the people who don't like GUI for the Galaxy, and it uses BioBlend library to submit the workflows to the Galaxy. So like it takes all the files, parameter, credentials, or like input files and etc. And it will upload the data set to the Galaxy. It will upload the workflow. It will run the workflow, and it will give you the results back. And the main benefit of it is like it can run on any Galaxy. It can run on your local machine. It can run on the cloud, on clusters, or like public instance if you have access to. And the more info on this thing on Galaxy Workflow Executor is available on GitHub. You can install using pip or conda. Like it's very easy to install and like start using. So like let's come back to the infrastructure. So like we have this deployment hosted at the BI Embassy Cloud. So like it ensures the security and the scalability, and like it utilizes the resources efficiently. And like it's been used actively for this consortium. Like we analyze like hundreds of samples already, and like we have more samples coming in every day. And also like it helps us like reducing the significant overhead time for like maintaining the cloud or like the cluster, and also like the turnaround time for the sample processing like is very efficient. So like we have two clusters deployed, like one development for all the testing and like developing new workflows, developing new tools, and also like we have a production cluster. Production is like a main workhorse. So development cluster is like it's very lightweight with one worker, with one master and the 2 worker nodes, while the production is like a 2 master and 3 worker node with high memory machines. So like these are the main cluster for analyzing all the single cell data. So benefit and outcome of this, so like we have a reproducible system. Like it can like if we don't like for any reason, like if we can't use our own if we can't use our own cloud, we can like move to Google or Amazon easily. Like also like a fast turnaround time, like every sample, because like it works 24/7. Like all the samples are analyzed like without us manually clicking on the buttons. And also like it's easy to scale for new users, new workflows, new tools, etc. And also the good thing it can serve as a model for any like collaborative project. Like if you have multiple institutes, multiple labs submitting samples and analyzing dataset, like this can be like adapted to other projects as well. So I'm almost done. So like this is a key takeaway. So like we have a automation and automation and the reproducibility are not an option in modern biology, but like they are essential. Like we must need it. And like our infrastructure is like it's meeting all these challenges that I like described in the beginning. And like this is like a successful example of the real life setting. And yeah, it can be adopted everywhere. And like for other projects. And yeah, that's it. Thank you too.
Thank you very much. Any questions?
I think I might have missed part of your presentation. Do you run the Galaxy instance on Codon or run it on the?
The Galaxy is on cloud.
On the cloud. Okay. Is it ITS maintained, a core facility maintained, or you maintain yourself?
Core facility maintains.
Oh, okay. Thanks.
One more question. Yeah, 2 questions. Why would you use Slack and not email? That's the one thing. And how many samples do you process per week?
So like we use Slack because like it's like all collaborators are on the Slack channels, so like they can monitor easily. Like we don't want to spam anyone who doesn't want to get notified. Like, for example, like if you have 10 collaborators, like if you submitted a sample, like you want to know, like other nights don't want to know what's going on with your sample. Like the Slack is like you can mute, or like you can just like to check. And there's, sorry, what was the second question?
How many samples do you have?
So it's like there's no fixed number. But like we check sample 24/7. Like whenever our collaborators submit the sample, we produce it like maybe couple of samples every day. So it's like maybe 10, 15 a week, because like they're really like it's very like the samples are very large. So like it takes like 7-8 hours to like process one sample. So it takes. Thank you.
Thank you very much.